Thank you for joining this webinar. It is a great pleasure to have today Ray presenting his work on this set of webinars that we have organized in Proteostasis. And I would like to thank Ray for two major reasons. Of course, for accepting the invitation, but also because where he is very early in the morning. So I'm sure that you know, he doesn't often give talks at 8 o'clock in the morning. So really appreciate his effort to do this, so we can combine everything at a reasonable time. I will just give a brief summary of Ray's extensive CV. He's currently in the Caltech Division of Biology and Biological Engineering Department in California, and Howard Hughes Medical Institute investigator. He did his biochemistry degree in Cornell University in New York before his PhD studies in University of California, Berkeley. And then it was followed by postdoctoral research in Mark Kistner's lab in the University of California before 1994, where he started as an independent group leader in Caltech, California Institute of Technology. And since then, the rest, I guess, is history. Since then, he's been there and developing all his exciting research. According to, you know, in addition, he also is the founder of many other enterprises and activities, especially the Proteome Exploitation Laboratory, where they're performing state-of-the-art proteomics-based mass spectrometry analysis there. And he's also founder of major new pharmaceutical enterprise companies as the Proteolytics and Clive Biosciences, where they develop inhibitors, especially on the P97 VCP ATPase and other components of the Ubiquitin system. And of course, I mean, Ray is one of the leaders of the field, and he has made major contributions in protein homeostasis and the Ubiquitin Proteasome system. Just to highlight a few of his, I guess, major discoveries in the area, it was on the cell cycle regulation, on the degradation of ubiquitination degradation of cyclin-dependent kinase inhibitors. He made important discoveries of how ubiquitin chains are recognized by the proteasome system, especially on the RPN11 adapter receptor protein. And another field, I guess, which his lab spends a lot of time at the moment, is how ubiquitin ligases are controlled. He was one of the co-discoverers of the kallin-based E3 ligases, which we now know they form the major or the biggest class of E3 ligases in the system. And his lab is focused a lot on understanding how these very complex E3 ligases are controlled, especially by this post-translational modification with a ubiquitin-like molecule NED8. And then, of course, another key discovery of his group was the identification of CSM5 as a key DNA-deleting component of the COP9 signalosome, which controls the activity of the CRLs. And another major activity of his lab at the moment, I guess, is the role of the ATPase P97 VCP, which is a non-ubiquitin receptor, which acts downstream of the ubiquitin E3 ligases. And a lot of the emphasis of his group now, which I guess he's going to talk us about today, is to develop specific inhibitors for these ATPases, and how he can combine these inhibitors with other inhibitors that are currently used within the ubiquitin system. So his work, of course, has been rewarded with many awards. And I would just mention the most recent one that he received, that he became a fellow of the American Association for the Advancement of Science in States, and also as a member of the American Academy of Arts and Sciences. So, and just to finish off, I will say that Ray is an excellent speaker, and I'm sure that you witnessed today. And just to recap the title of his talk is Targeting the Ubiquitin Proteasome System in Cancer, the Proteotoxic Crisis Hypothesis. Again, thank you very much, Ray, for doing this, and we're all going to sit back and enjoy your presentation. Thank you very much.
Thank you. That was that was a very generous introduction. Without further ado, I'll launch into the topic of the talk, and I assume that many of you in the audience are pretty familiar with the ubiquitin system, but in the event there's some who are not, this just shows a very general organization of the system, which will be the focus of my talk today, where you have an E1 enzyme that activates ubiquitin for transfer to proteins. It's transferred to an E2 enzyme, and then working with, okay, wait a second now, I had a trouble, my animation worked earlier, and now it's not working. Uh-oh. Okay, we still there?
Yeah.
Yeah, we're still good.
Yeah, I don't know why. Now it's working all of a sudden. Okay, so the E2 ubiquitin conjugate works with an E3 ubiquitin ligase enzyme to transfer ubiquitin onto substrate proteins. And one very well-studied famous example is ubiquitination of cyclin protein to inactivate cyclin CDK. And that's an example of a particular form of ubiquitination where it's used to regulate a process, in this case to shut off CDK enzyme by degrading the cyclin subunit of the complex. And this form of regulation is a very well studied means by which the ubiquitin system is used, and there's literally hundreds to thousands of substrates that are regulatory substrates of the ubiquitin system that are used to control cellular processes. In addition to that, the ubiquitin system is used in a completely different way as well to essentially perform quality control. And so as genes are expressed, there's constantly mistakes occurring in transcription, during translation, as well as during folding and assembly of the polypeptides. And if you end up with a defective polypeptide, as shown here, those polypeptides have to be eliminated from the cell, and that most commonly employs, again, the ubiquitin system to ubiquitinate and then the proteasome to degrade those substrates. And this affects essentially the entire proteome because all proteins are going to be produced at some level in a defective state. Now, there's many components in the system. There's on the order of 1,000 of them, and the E3s are the biggest class of components, and they represent a key node for regulation and specificity of the system. Now, this system is extremely important for human health. There are mutations in a number of genes of the system that cause devastating human disease, and I discuss quality control. Mutations in components that are involved in quality control, and Parkin is one example, are known to lead to human neurodegenerative diseases. On the other hand, mutations in the enzymes that are involved in regulation, and the FBOX protein FBXW7 is an example, predisposed to cancer. And FBXW7 is one of the major human tumor suppressor genes. And so this is really fueled interest in this area to understand how this plays an important role in human health. And what I'll tell you today is that quality control, in addition to being important to stave off neurodegeneration, turns out to be also important, we believe, for the biology of a number of different types of cancer cells. And that may be something we can exploit to develop new inhibitors for treating cancer. Now, the idea I just put forward there is illustrated graphically here. And what I show you on the left-hand side is what I imagine is occurring in a normal cell, where you have the proteasome representing the ubiquitin proteasome system, and it's an appropriate balance with its substrate proteins, with the load that's presented to the proteasome. And this balance is obviously established over hundreds of millions of years of evolution so that cells have an appropriate amount of UPS activity to deal with the substrates that they are going to be generated. On the other hand, in a cancer cell, that balance can get out of whack. And it can get out of whack for a few different reasons. One being that some cancer cell genomes are littered with mutations. So lung cancer and melanoma, for example, can have over 1,000 coding sequence mutations per genome. But perhaps even more important, a very large fraction of cancers, it's been estimated by Don Cleveland that it's over 90% of solid tumors, are highly aneuploid. meaning that they have other than a 2N chromosome number, usually due to gain of chromosomes. And those extra chromosomes remain biologically active. They encode proteins, which are synthesized. And to the extent that those polypeptides form complexes, which many polypeptides do in eukaryotic cells, it will be out of balance with its partners. So for example, if you have an aneuploid chromosome that encodes several ribosome subunits, those subunits will be made in excess, and the way the cell deals with that is to degrade those excess polypeptides. Now, that puts an additional tax on the ubiquitin system. There's more proteins in a cancer cell than the system was evolved to handle. And what that means is that the cancer cell is potentially more sensitive to inhibition of the system because it has a higher demand for activity of the ubiquitin proteasome system. And that is what I'm hoping in this talk to see if we can develop mechanisms to try to exploit that potential difference in demand in a cancer cell versus a normal cell. So the question is, it's an intriguing idea. Is there any evidence that this idea is correct? And probably the best piece of evidence comes from the FDA approval of bortezomib, which occurred in 2003. And bortezomib is a proteasome inhibitor, and together with the immunomodulatory compounds of the thalidomide family, it has really transformed patient care in multiple myeloma, which is a really terrible form of cancer that arises in about 15,000 people in the US alone every year. Now, bortezomib works great in multiple myeloma and another cancer, mantle cell lymphoma, where it's really changed the prognosis for patients. But as you know, one problem is that it only works in those diseases. It has not shown efficacy in other cancers. And so around 2003, this spurred Craig Crews, who's at Yale, and I to form a partnership and start a company. We started a company called Proteolix to test the hypothesis that perhaps the narrow range of action of bortezomib was because the compound itself maybe wasn't potent enough or didn't have enough durability, good enough pharmacokinetic properties, and that if we could make a better proteasome inhibitor, if you will, that potentially we might be able to expand into other cancers. And oh my goodness, this thing is, I'm having trouble with my animations. So my computer seems like it's going to sleep on me, but we're back. So in fact, it has turned out to be the case. Carfilzomib demonstrated, carfilzomib was developed by Proteolix, and that compound has demonstrated clear superiority over bortezomib in a clinical trial in relapsed myeloma patients. So they doubled the progression-free survival shown here. But despite that success, it is yet to demonstrate efficacy in other cancers, much like bortezomib. And so that led me to ask a couple of questions. The first is, maybe inhibiting the 20S proteasome, which is what bortezomib and carfilzomib target, is not the right way to inhibit the proteasome. Maybe there'd be a more effective way to inhibit it. Alternatively, maybe the proteasome is not the right target, and maybe there are other targets in protein quality control that we can hit that might provide a broader therapeutic benefit. And so those are the questions I'm going to try to address in the rest of this talk. So I'll first start with alternative ways of inhibiting the proteasome. So this is a cryo-EM image of the proteasome that I believe is from one of Andreas Martin's papers. And the 20S here is a cylinder, like, let's say, an empty paper towel roll. And on the inside of that cylinder are the active sites of the 20S proteasome that are inhibited by bortezomib and carfilzomib. Now, that's a key part of the proteasome, obviously, but in addition, there's this other structure, the 19S cap, that is required to get proteins with ubiquitin chains into the proteasome where they can be degraded. And there's a number of activities in that cap that are potentially inhibited to block the proteasome, including these ATPase activities, RPN13, which is a ubiquitin receptor, and other labs have worked on. But we were particularly intrigued by RPN11. And RPN11 is an enzyme of the proteasome that my lab discovered, oh, I don't know, about 13, 14 years ago now. And RPN11 is a zinc metalloprotease. And what it does is it cuts ubiquitin off as a substrate is being inserted inside the proteasome. It's absolutely essential for proteasome activity. And so we reasoned that that might be an interesting target for inhibition. And why we think it might be a good target is we thought it could potentially have a more profound effect on proteasome activity. And the rationale is shown here. So the proteasome has 3 active sites on the inside. And if you treat with bortezomib or carfilzomib, they predominantly inhibit one site, the chymotryptic site, with much less activity on the other 2 sites. And so when you inhibit the proteasome with these compounds in a human, you're really not shutting down all of the protease activity of the proteasome. On the other hand, RPN11 is required to cut ubiquitin off as proteins are being inserted into the proteasome. And if you block RPN11, then the idea is the substrates will no longer be able to fit inside the proteasome. The RPN11 will serve as a cork to plug up the proteasome, so the substrate doesn't even reach the active site. And we reason that this could potentially provide a more profound form of inhibition. Now, an important question is, can RPN11 really be targeted successfully? And we reasoned that there was a good chance it could be. First of all, because it's an enzyme of a functional class. It's a protease, and these have been successfully targeted many times in the past, including the proteasome inhibitors themselves, ACE inhibitors, HIV protease inhibitors, and so forth. And second, it belongs to a family of hydrolases, zinc metallohydrolases, that have also successfully been targeted in the past. And a very famous example would be the ACE inhibitors, the angiotensin converting enzyme inhibitors like captopril, and there's a whole family of these inhibitors that are used to treat hypertension, HDAC inhibitors, among other compounds. And so we reasoned this should be a druggable target. And so this challenge of trying to drug it was taken up by Frank Parlati. He spent a sabbatical in my lab from industry and then returned to industry and is now at Calithera Biosciences up in the Bay Area. And what Frank did is he set up an assay for measuring RPN11, where he produced a fusion of 4 ubiquitins in tandem, fused to a 30 amino acid peptide with a unique cysteine that he could label with a fluorophore. And when you add the proteasome, what it does is it cuts the peptide off of this ubiquitin chain, and in so doing, it changes the fluorescence polarization. So you start with a very high polarization for the substrate, and as you convert it to product, that small peptide has very low polarization, so you see the polarization drop during the reaction. If you add a conventional 20S proteasome inhibitor that blocks the chymotryptic site, epoxamycin, you'll see it has virtually no effect in this assay, which is exactly what we would expect, since this is all going on the outside of the proteasome, in the proteasome lid. If we add ATP gamma S, that inhibits this activity, and that's consistent with findings we reported previously that RPN11 activity requires ATP hydrolysis. Likewise, if we add O-phenanthroline, which is a very commonly used broad specificity zinc chelator, that also wipes out the activity. And that and other lines of evidence indicate that this activity is really due to RPN11. So using that assay, Frank then undertook 2 efforts to do 2 different screens. The first screen was what I call an extremely low throughput screen that was focused on a collection of metal binding compounds that was developed by our collaborator in this effort, Seth Cohen, who's the professor at UCSD in San Diego. And the other effort was we engaged the NIH Molecular Libraries program to do a conventional high throughput screen, and that was done at Sanford Burnham. And this just shows the sort of summary results of the screen. Many compounds were detected in the first instance. We did a number of triage screens to find ones that were specific for RPN11 were not inhibiting other zinc enzymes and were effective in cells. And at the end of the day, this led us to 2 compounds. And what's remarkable, so 1 compound from the low throughput screen and 1 compound from the high throughput screen. This was essentially obtained from 100 compounds. This was obtained from 350,000 compounds. What's remarkable is despite that huge disparity in screen size, they are essentially the same molecule. The only difference is that in this case, the core molecule shown here has been thioesterified with this R group right here. So when you put this molecule in water, it hydrolyzes to produce this molecule. Now, I can tell you if I'm given the choice of screening 100 compounds or 350,000 compounds, it's an easy choice. Anyhow, both of these molecules, as you would expect, have a essentially the same potency with an IC50 in the range of 1 to 2 micromolar in the RPN11 assay. And these are very, very specific. We believe they are inhibiting RPN11. There's a number of lines of evidence for that. One that I'll show you here is we had a compound known as cyclin, which is a bioinorganic zinc complex that chelates zinc much the same way that an enzyme active site does. And when we add that compound to our reactions, you can see it shifts the IC50 curves over to the right by over an order of magnitude. So it's essentially just mimicking the active site of the enzyme, and it's titrating out the inhibitor. Okay, now to try to figure out how this compound was inhibiting our active site, first we had a hypothesis, and the hypothesis was that this nitrogen and the thiol were essentially acting as a chelator to form, each of them forming a ligand with the zinc. And if that was true, we should be able to do an experiment that any molecular biologist is familiar with, which is essentially to do a deletion or mutation analysis on our compound. And so Mathieu Ruffet and Seth Cohen's lab removed the nitrogen here, moved it up to the top of the ring, put a methyl group on the sulfur, changed it to a hydroxyl group, or changed it to a methyl group. And all of those modifications essentially killed the enzyme activity. Now, one thing I haven't told you is this compound also inhibits other members of the so-called JAMM family. CSN5 is another member. All told, there's 7 proteases in this family. And you can see we also have activity on CSN5, and that behaves similarly in this SAR analysis of the compounds. Cytotoxicity tracks with that, except this compound, which is also quite cytotoxic on cells, but presumably for a different reason. Okay. Oh, I'm having that same problem again. Let's see. There we go. Okay, so the next experiment we did is we wanted to test, let me go back here, how pliable this scaffold was. Can we modify it at all these different positions to try to dial in more potency or specificity? And this shows that the compound is actually quite pliable, that we can modify it at the 3, 4, 5, 6 positions and essentially have no effect on the RPN11 activity. We even get some specificity when we modify on the 4 position here, where now we get more specificity towards RPN11 over CSN5. And an important point of this slide is that these compounds are quite selective for these JAMM proteins. We looked at a few other zinc enzymes, so HDAC6 and 1, deacetylases, matrix metalloprotease 2, which is a zinc metalloprotease, and carbonic anhydrase, which is also a zinc metalloenzyme. And all of these are either not inhibited at all or inhibited only very, very weakly by these compounds. 5-lipoxygenase is an iron-based enzyme, and it's also it's not inhibited at all, essentially. So these compounds are quite remarkable. They inhibit the JAMM proteins, but they do not appear to inhibit other classes of zinc hydrolases. And conversely, when we got inhibitors of other zinc hydrolases, they did not affect the JAMM proteins. So there's something very special about the active site of these proteins. So to dig more deeply into this project, it was taken over by Jing Li, who's a postdoc who's currently in my laboratory. And this just shows a summary of a lot of effort that was carried out by largely by Christian Perez, David Puerta, and Mathieu Ruffet in Seth Cohen's laboratory. And after synthesis, I don't know the exact number, maybe we're up to about 130 compounds now. The best compound we've come up with is this molecule 3027. And you can see we've gotten significantly better in terms of potency. Importantly, it's gotten much better in terms of specificity for CSN5. And this was the most difficult thing here is AMSH, which is another family member of the JAMM family. And you can see the original molecule was more potent on AMSH than RPN11, but now after the optimization, we have about tenfold more potency on RPN11. And this compound also works very nicely in cells to block degradation of a substrate, a reporter substrate, UBGFP, that's commonly used in the field and whose degradation requires the proteasome. And this just shows you some of the, there's a lot of data we have on this compound now. I'll just show you a few pieces of data in the interest of time. So this just shows cells with UBGFP. If you treat them with MG132, which is a proteasome inhibitor, they go from being dark to being bright green. And likewise, 3027 has a very similar effect on inducing accumulation of this reporter protein. 3021 is just an earlier generation compound. And likewise, we can see an effect of these compounds on formation of agrosome structures that are labeled with ubiquitin in red here, HDAC shown in green, or P62 also in green, the autophagy regulator. And these are driven by MG132, and likewise 3027 causes a very similar effect on inducing formation of agrosomes. And yet another type of analysis, we've just looked at ubiquitin conjugates, and you can see as we titrate up 3027 here, that we go from a normal basal level of polyubiquitin conjugates to an elevated level, about 2 to 3-fold higher than the basal level, very similar to what we see when we use a conventional proteasome inhibitor. And this shows here that NRF2, which is very frequently used as a marker of proteasome inhibition, is accumulating as we titrate up the RPN11 inhibitor 3027. And this just shows that the inhibitor, consistent with the biochemical data, it does not inhibit the CSN in cells because it does not cause accumulation of a netylated form of the CUL1 protein, which would occur if it was inhibiting the signalosome. And these are very brief treatments. This is just 2 hours of treatment. Now, to dig more deeply, I'm sorry, let me back up. To dig more deeply into this result, where we see these polyubiquitin conjugates, we wanted to know what are the proteins that are accumulating here. And so we did what's called a gly-gly analysis, where you treat a cell lysate with trypsin, and any peptide that is ubiquitinated in the cell will now have a diglycine remnant from the ubiquitin that's attached to it. And there's an antibody that we can purify those peptides and then analyze them by mass spec. And this shows a volcano plot where the x-axis gives you the ratio of that gly-gly peptide when we inhibit with 3027 compared to DMSO. And the y-axis gives you the p-value. And the higher up on the axis, the better. And so in this analysis, we saw many proteins that are accumulating as gly-gly peptides. And we did the same experiment with bortezomib, and we saw very similar results with bortezomib. But one thing that was really interesting is this one protein here, angiomotin, where we saw very strong gly-gly peptides from angiomotin with the RPN11 inhibitor, but not with bortezomib. And so this just shows a validation of that result, where if we look with anti-ubiquitin staining, cells treated with 3027 or bortezomib, you see high molecular weight ubiquitin. These purify on a tube resin. And if we now blot for angiomotin, you could see high molecular weight conjugates in the lysate, or after purification, with 3027 inhibition of RPN11, but not with bortezomib inhibition of the 20S proteasome. So this suggests there might actually be something different on a molecular level about inhibiting RPN11 compared to inhibiting the 20S proteasome. And this is something we're very, very interested in pursuing further, because it could potentially lead to a clinical differentiation of RPN11 inhibition from 20S proteasome inhibition. Now, the final piece of data I'll show you in this part of the talk, we just set out to try to develop some genetic evidence that the target we're really inhibiting is RPN11. And we've tried unsuccessfully to isolate resistant cell lines. We get resistant cell lines, but the resistance is not stable. And so we cannot really characterize the cell lines because over the course of a month, the resistance is lost. But instead, what we did is we used NRF1 knockout or wild type MEFs. And we had shown previously in a paper in 2010 that these MEFs, when you knock out NRF1, become more sensitive to 20S inhibitors because NRF1 promotes the expression of the proteasome in cells. It promotes high-level expression. So when you lose NRF1, proteasome expression diminishes a little bit, and cells become more sensitive by about 2 to 3-fold to a proteasome inhibitor. And likewise, they become, you can see here, about 2.5-fold more sensitive to the inhibitor of RPN11. And another experiment we're doing currently with Martin Kampman at UCSF is he's shown that his CRISPR-I system, where he can knock down 19S subunits, once again sensitizes cells to our RPN11 inhibitor. And so based on these lines of evidence, we believe that the activity that we have is on target. And so this is very early days. There's much more to be done, but I'm very optimistic that we will be able to develop these compounds further and potentially, if we're fortunate, take them forward into the clinic. Okay, I'd like to switch gears now and talk about P97. And, you know, this fits in with, in the slide I showed earlier, that one idea was maybe the proteotoxic crisis, we're not seeing broad inhibition of cancer cells by the current proteasome inhibitors, either because maybe it's not the most favorable way to inhibit the proteasome. The other idea was maybe the proteasome is just not the right target. And so we wanted to pursue that idea further. And so, you know, we were thinking about what target should we aim at in protein quality control. And we were drawn to P97 because it's thought that one of the key mechanisms of action of the proteasome inhibitors like bortezomib is that they block this process known as ERAD, which stands for endoplasmic reticulum associated degradation. And in that process, if you have a membrane protein that misfolds, maybe due to mutation, it's retrotranslocated into the cytosol where it meets the proteasome and is degraded. But if you inhibit the proteasome, it accumulates in the cytosol and it can trigger an irresolvable unfolded protein response that then leads to apoptosis. And this is thought to contribute to the mechanism of action of bortezomib in multiple myeloma. Now, when we were thinking about targets, we looked at this guy here, P97, also known as VCP, and this essentially serves as a motor that reels the protein into the cytosol across the ER membrane. And we reasoned if we inhibit this, maybe we could gain many of the benefits of inhibiting the proteasome, but perhaps with much less of a side effect, because this has a much narrower range in the UPS than the proteasome does, and suggesting that maybe we could inhibit it much, much harder and for longer with less unfavorable side effect. And so the way I'm thinking about this when we set out on this project is illustrated by this slide here. So you could think of the ubiquitin proteasome system as a funnel, okay, where each of these colors represents a different pathway in the system. And this is simplified because you really need about 500 of these colors to fully represent the system, but then this diagram would be hard to interpret. So let's just imagine these are all our different pathways. We inhibit the proteasome. It's like sticking a cork in the bottom of the funnel. It blocks all of these pathways which converge on this central choke point that is the proteasome. On the other hand, if we knock out P97 activity, we should only hit select pathways in this target funnel, because not all pathways require P97 function. And in fact, if these pathways are enriched for quality control pathways, all the better, because our hypothesis is that it's really the quality control function that's important for cancer cells, their reliance on the UPS. And so what does P97 do? This just illustrates a number of the functions that have been figured out over the past few years. So I've already told you about ERAD. A few years ago, my lab, Jonathan Weissman's lab, showed that it's also involved in this ribosome-associated degradation process, and that's another quality control mechanism. It's been linked to autophagy, although the molecular mechanisms here are not well understood. It's been linked to the handling of protein aggregates. It's been linked to mitochondrial-associated degradation. All these pathways, the MAD, the aggregates, the autophagy, the ribosome, the ERAD, these are all quality control pathways. So P97 has a very, very prominent role in cellular quality control. And so this made it an even more attractive target to us. Now, what P97 does in these pathways, at a biochemical level, we still don't understand it very well. But the general thinking is that if you have a substrate that's bound to a biological structure, and that could be a protein complex, a membrane, a chromatin, and that protein is modified with ubiquitin, P97 can be used to extract it from the biological structure. And once it's extracted, perhaps also help unfold it to thread it into the proteasome for degradation. And an important point is that there's only a subset of proteins in the UPS system that require this activity, unlike the proteasome, which is required to degrade all of the substrates of the UPS. So the idea of pursuing this was then taken up by Tuifeng Cho, who was a postdoc in the lab at the time, and she's now a faculty member at LA Biomed, which is part of Harbor UCLA Medical Center, not far away from here in Long Beach. And so she set out to develop an HTS assay where she could measure in high throughput P97 ATPase activity. And that assay then was the basis for doing a high throughput screen. The high throughput screen was carried out by Hugh Rosen's group at the Scripps Research Institute, and they were part of the molecular libraries program. And that led to this molecule here, DBEC, which is, you know, about a 1 to 2 micromolar ATPase inhibitor, and it blocks the turnover of UBGFP, which we know from the work of a number of other groups is a P97 dependent substrate of the UPS. Now, this was an initial hit. It was a reasonable potency, but we wanted something to see if we can get something a lot better. And so we engaged this chemistry group of Frank Shainin at Kansas University. They were also part of the NIH Molecular Libraries program, and they bought about 60 analogs of this, and they synthesized another 140. And of the compounds that they synthesized, we identified this compound here, ML240, as being the best compound in the series. And this was about 10 times better than our starting compound, DBEC, a little bit better, a few-fold better in the cell-based assay, but still, this compound was not good enough to be a drug. The potency is reasonable, but it was not a particularly soluble compound. It's rapidly metabolized by liver microsomes, and it binds with pretty high efficiency to proteins in human plasma. And so we reasoned this would not have the proper pharmacokinetic properties to be a good inhibitor. And, but we had already made 200 compounds, which is a pretty big effort in an academic SAR exercise. And so to push the ball further, I reasoned that we really needed to enlist some serious professional help on this, some serious drug development expertise. And so together with Seth Cohen, who we were collaborating with on the RPN11 project, Seth and I went out and we raised venture capital to start Cleve Biosciences. And then they really went to town on this scaffold. They made another, I think, well over 500 compounds now. And out of those compounds, the best compound to emerge was this compound here, which is called CB5083. This is again about 5 times better in potency than our compound, a couple-fold better in a cell efficacy assay, but more importantly, it's much more soluble, it's much more resistant to liver microsome metabolism, lower plasma protein binding, and overall just much better pharmacokinetic properties and properties consistent with being a candidate drug. So this compound then, and if you're interested in more details on this, they published it in Cancer Cell, I believe in the November issue, where they published all of their preclinical data. And this just shows an example of this compound's efficacy. So this is an A549 xenograft. This is a lung cancer from human implanted into an immunocompromised mouse. And you can see if you don't add any drug, this grows rapidly in the mouse over the course of 20 days. And if you try to treat it with the existing approved proteasome inhibitors, so the purple is ixazomib, which was recently approved. It's an oral version of bortezomib. The green is bortezomib. The blue is carfilzomib. And you can see there's some activity of these inhibitors on the A549, but relatively modest activity. And importantly, these are being dosed at their MTD. So we're dosing these as high as they can go. Now, the red, on the other hand, shows CB5083, which shows almost flatlines this A549 xenograft. And this is but one result. Cleve has seen this with at least 2 other solid tumor models that are reported in their Cancer Cell paper. And so, you know, I think results like this build considerable optimism that inhibiting quality control by this mechanism may provide a more profound inhibition of quality control that might now enable us to see efficacy in solid tumors. Now, on the strength of these preclinical data, Cleve filed an investigational new drug application with the FDA, and that in turn has led to clinical trial. They're currently in 2 phase 1 clinical trials. They've been dosing humans for, close to about 1.5 years now, and they are well along in their phase 1 trial, and we are eagerly awaiting to see how those results come out. But on the strength of results like this, I'm very optimistic that we may have a way to really test this proteotoxic crisis hypothesis in a rigorous way in human disease. And I guess the answer is, at this point, is stay tuned. Now, just to tell you what I've told you in this talk, we've developed 3027 as a first-in-class RPN11 inhibitor. It works by binding to the active site of RPN11 via its catalytic zinc atom. 3027, like 20S inhibitors, causes accumulation of polyubiquitinated protein, but has a differential effect on at least one substrate, and we expect there's probably more substrates like this that are going to be selectively affected by this inhibitor. We just haven't characterized all of them at this stage. Number 3, then I turned to talking about P97 after RPN11, and I described the discovery of DBEC, which is a first-in-class AAA ATPase inhibitor, and it blocks multiple quality control pathways and induces a strong unfolded protein response by inhibiting P97 VCP. And finally, Cleve Biosciences took our scaffold ML240 derived from DBEC. From that, they developed CB5083, which is a first-in-human P97 VCP inhibitor that, again, blocks multiple quality control pathways and induces a strong UPR, and that is currently in human clinical testing. And drug discovery is a very, very difficult, very time-intensive, very technology-driven process, and none of this would have been possible without very important collaborations that are indicated on this slide. The Proteome Exploration Lab helped with the RPN11 glide proteomics. Seth Cohen's group at UCSD helped in the development of the 3027 compound. The Sanford Burnham HTS group, led by Dr. Thomas Chung, did the HTS for the RPN11 thioester scaffold. The TSRI group did the HTS for identifying DBEC, and Frank Shainen's group at University of Kansas did the SAR optimization of that scaffold. And thank you for your attention, and I'd really be happy to take any questions you might have.
Thank you very much, Ray. That was a great discussion. If you do have any questions, we'll open the floor to questions. If you could type the question that you'd like to ask in the question pane of the GoToWebinar window, we can then select you and open up your microphone if you have one, so you can ask it directly to Ray. If you don't have access to a microphone on your computer, that's fine. We can ask the question for you. While we're waiting for you to frantically type out your question, I believe Dimitris, did you have a question?
Well, yeah, I mean, I can, start off. So, I mean, thank you very much for this wonderful presentation. I was just wondering, I mean, you mentioned a lot that during the proteotoxic crisis, of course, the proteasomal activity is blocked and the system upregulates components of the proteasome. Is it anything known what happens with the activity of the E3 ligases under these conditions? Do they work better or worse? Do they start to, I don't know, ubiquitin substrates much more because they accumulate and they feel that they have to be eliminated? So is anything known about this?
You're saying in response to inhibiting cells with a proteasome inhibitor, or just in cancer cells in general?
Well, I mean, I guess in both cases. So when the system, when you start getting accumulation of the load, as you mentioned, in cancer cells, when you block the proteasome, how does the E3 ligase system respond to this? I mean, do you think that there is any increase in this activity to start ubiquitin substrates much more, or there's no any effect?
To my knowledge, that has not really been investigated in any detail. Most of the effort to look at, say, transcriptional responses or gene induction responses when you induce a proteotoxic crisis, for example, with a proteasome inhibitor, have really focused on expression of the proteasome itself, right? Because this was shown many years ago. Elke Kruger's lab was one of the pioneers in showing that. It was also shown by Ken Anderson in human patients treated with bortezomib, that you have this broad transcriptional response of the proteasome inhibitors. And so a lot of effort is focused on studying that response mediated by NRF1. I'm not familiar off the top of my head with similar studies looking at ubiquitin ligases that might be induced by that response. There's many different quality control pathways, and those ligases tend to be constitutively expressed because there's a constant burden of misfolded proteins. But whether there's a further upregulation, I just don't think that's really been investigated in detail.
Okay, we have a question from Timo Kurt. I apologize if I've mispronounced your name. So I'm just going to unmute you, Timo. Can you hear me?
Yes, I can. Can you hear me?
Yes, I can. Please go ahead and ask your question.
Hey, Ray, does CB5083 inhibit other AAA ATPases? And do you guys know the mechanism of action?
So, hi, Timo, by the way. So CB5083 itself, DBEC inhibits at least one other AAA ATPase. It's the, what is it, VPS 4, I think it is, the one that's in the escort pathway. But CB5083 does not, as far as we know, but not every AAA has been tested. So what we've done is there's a panel that's maintained by Activex, a company in San Diego, where they have an assay, a mass spec-based assay. And in that assay, CB5083 did not inhibit any other ATPases. it showed very weak inhibition of DNA protein kinase, but it was more than an order of magnitude. I think it was 2 orders of magnitude less potent on that than it is on P97. In terms of the mechanism of action, it's a competitive inhibitor. It's ATP competitive. And so it appears to dock in the active site. And Cleve has isolated resistance mutations using the Turin-Kapoor method of HCT116, mutator cell lines, and looking for resistance. And they get resistance mutation peppering the D2 domain of P97, which is the domain that has most of the ATPase activity. And so those data support the biochemistry, which indicates that it's a competitive inhibitor of the ATPase activity of that domain.
Right. So from that answer, I take it you don't have any structures yet?
No. So my lab, in collaboration with a number of other groups and Sriram Subramaniam at NCI, recently published a non-covalent, a non-competitive inhibitor that's docked on P97. It was done by cryo-EM. And that compound is not bound in the active site. It's probably bound in a similar position as the NMS873 compound that was reported by Nerviano and Genentech. Subramaniam, as far as I know, is collaborating with Cleve on doing the same thing with CB5083, but that's a work in progress.
Right, okay. Can I ask one more question?
Yes.
If it's a quick one, yeah.
Yeah, just a real quick one on the RPN11 inhibitor. So I'm wondering a little bit about the logic behind this, because you were mentioning that, you guys go after it because you think that, OMIBs are not very good inhibitors of the proteasome, and that you maybe, get a better result with RPN11. What's the current thinking about, I always thought that one reason why, bortezomib doesn't work on solid tumors is because of the pharmacokinetic properties, that it's just simply cleared too quickly, which is also one way that allows it to actually work in humans without being too detrimental. Is that still the current thinking, or do you think that's not right? And that, in fact, if you were to inhibit better, and if you were to be able to, penetrate into tissue and stay around longer, you'd actually have a therapeutic window to achieve, anti-tumor effects without, having all these negative effects on, the healthy cells?
Well, first I want to walk back one of your comments there. The OMIBs are really great inhibitors, right? They are
of the chymotrypsin side, yeah.
And carfilzomib is extraordinarily specific, but they really inhibit the chymo site, and so they don't necessarily wipe out all of the protein breakdown activity. They really wipe out the chymotryptic site activity. Now, why might RPN11 be different? One key thing I really didn't get into in the talk, you'd mentioned, is that the PK of the OMIBs is not very good, right? They are, when you inject them into a person, they are cleared from the plasma within minutes. And they have a very, very slow off rate. So bortezomib, I think T1/2 of 110 minutes. Carfilzomib, completely irreversible. So they hop on the first proteasome they see, they don't come off again, and so you don't necessarily get a good biodistribution throughout tissue. And that was the whole idea behind ixazomib, a new oral inhibitor that's a bortezomib-like molecule. And the argument there is it may distribute through tissues better. Whether that translates into better clinical efficacy or broader clinical efficacy remains to be seen. Now, with the RPN11 inhibitor, unlike the OMIBs, it's not a peptide. And we know it's orally available. There's already been studies in the mouse. And the hope is that if we can develop a molecule with more traditional PK for a small molecule, so a T1/2, let's say on the order of 2 or 4 hours, we can really slow down the recovery of proteasome activity and maybe achieve a more stable, durable inhibition of the proteasome, something that we have more capability of dialing in, if you will, and potentially better biodistribution as well. So I think you're hitting the nail on the head that PK may really be the way that we can improve on the current mode of inhibiting the proteasome.
Okay, thank you, Timo, for your questions. We have one from Giovanna Serino, so I will just unmute. Giovanna, are you there? Giovanna, can you hear us? It sounds a little...
Can you hear me?
Very faintly, so we'll see if we can, so ask your question.
I can hear her well.
Yeah, okay.
Yeah, I can, you know, there's always the problem that you inhibit one thing while promote other things, especially when you treat an entire organism with UBS inhibitors. So what are going to be the effect on neurodegeneration of these inhibitors? Are they going to have any effect, or there would be any interplay?
You know, at this stage, so the proteasome inhibitors, for example, they don't cross the blood-brain barrier. The P97 inhibitor shows very much lower penetration into neural tissue. The RPN11 inhibitor at this stage we can't say, because we don't really have the clinical candidate in hand. So, you know, I think will be something we'll have to look at just to see if it gets in the brain at all. Then that's something one has to potentially worry about. Now, you know, I would say on the flip side of that, with an oncology indication, you know, neurodegeneration is something that typically takes place over decades. When you have a mutant P97, you can get IBM-PFD, but it takes typically on the scale of 30 to 40 years for symptoms to develop. So, you know, I think that's one thing to bear in mind, that in an oncology indication, most of the people you're going to be treating are unlikely to have 30 to 40 years of life left. So, you know, it is something that does need to be thought about. And I think, you know, when we get to that point with RPN11, it's something we'll just have to look at.
Okay, thank you very much, Giovanna, for your question. Well, we're just about out of time right now. We're up to our hour. So I'd just like to thank our presenter today, Ray, for his time and for giving us an excellent webinar. I'd also like to thank all the organisers at the COST Proteostasis Network. We'll continue working together to bring you new and insightful webinars. We will have recorded this webinar, so what we will be doing afterwards is putting up on the Bio-Techne YouTube page, and we will shortly be sending you an email where you can go and see this webinar again if you want to. So all that leaves me to do is say thank you very much. I hope you enjoyed the webinar, and have a great day. Thanks very much.
Bye.
Bye-bye.